All right, let's get into some cell communication and some cell signaling stuff here. So, why do cells communicate? What does cell communication look like, and what are the different types of communication? Is what we'll be going through. This is kind of an important slide. You'll notice that there are three phases to signal reception, so those are going to be important for you. So, you have reception, you have receiving the signal, which happens right here, and you have transduction, which is moving the signal from the outside of the cell to the inside of the cell, going this way. And then you have whatever that response is going to be, right? Dealing with whatever that message was, and having the cell jump to action and some way. So those are really going to be the phases. We'll talk about this word a lot over here. We'll talk about ligand quite a bit today. So a ligand is a general term. It just means whatever the receptor is going to receive, and it can come in different forms. It can be a hormone, it can be an environmental stimulus, it can be an ion, it can be another protein, it can be lots of different types of molecules, but it's just whatever is bringing the signal to the receptor, right? And the receptor is this big purple protein that's lodged in the cell membrane right here, which is the big yellow thing, okay? So moving on here. So we have some local signals, and we have what's called paracrine, and then we have synaptic signaling as well. So over here, paracrine signaling down here. Para means adjacent or next to, so you have this cell that's releasing a lot of signals all at once, and it's going to target all of these cells that are adjacent to it. And so that's going to, be the ligand, a bunch of ligands for whatever the message is meant to do. And over here we have synaptic signaling, which works a little bit differently. Similar idea, but this is basically for your nervous system, how this works. So this is how the signal gets transmitted from one to another. So there's an electrical signal that comes down the neuron first, and then that triggers the chemical signal or the neurotransmitters, that word right here, the chemicals that cross the gap, right? The chemicals that cross the synapse, okay? So the synapse is the gap right here between one neuron to another. So the chemicals cross the gap, and then that will stimulate this target cell, and it might just be another neuron, and then another signal will carry on down that one, and so on and so forth. It'll kind of keep going electrical, chemical, all the way down the line. A few different ways that molecules also move in between cells, especially adjacent cells. So some of them they kind of all do the same thing, just have some different names. So gap junctions are what we tend to call the animal cell gaps, or wherever there are some specific gaps in the membranes that butt up to next to each other. We call those gap junctions, so that molecules can go back and forth pretty easily. Same idea in plants, but we call them this fun word, plasmodesmata. Okay, plasmodesmata are like tunnels in the cell wall that will allow molecules to go back and forth between plant cells. And then down here we have what's called cell-to-cell recognition. So you have kind of like a ligand attached to a cell membrane, and then you have the receptor on the other one, and they'll only fit together like puzzle pieces. That's how you know if it's actually going to fit. Many of them won't fit, and so they won't bind to each other. They have to be specific size and shape, structure and function. Long distance signals, we'll discuss a little bit more in detail down the road. And actually, we don't have to anymore. That's more endocrine system stuff, which they actually pulled the endocrine system out. But we still may mention it down the road in the spring when we get there. So endocrine down here, hormones, and they travel long distances. So really what that means is that you're going to use the bloodstream. You're going to use the bloodstream to move things all around the body long distances versus just like, around, instead of the cells, right around you, okay? All right, so we get into some cell communication here, and now we're going to get into some more specific pictures and more specific examples. So the signaling molecule here, the green thing, also ligand, right? That would work. And if you notice, the receptor, this big yellow Y-looking thing, so the receptor is embedded in the membrane. And notice down here, this is unactivated. And you'll see this little tail is curled right here, and he's unactivated. And then what happens is you have the ligand come and bind, and then that causes a conformational change on the inside. And now instead of being, little hooked that way, it straightens out. And so that's going to become more available for molecules like this to come and like bind here, or maybe that's and then cause a signal to go in this way. So that's one way it can happen. We'll look at a few different ways. So remember the three phases. You have the receptor activation, which happens out here. Then you have the signal transduction, which is kind of a cascade, is what we call it, a cascade of events, where it's kind of like, playing that telephone game where you pass the note. And so the signal just keeps getting passed from one molecule to another. And then you have your cellular response, which really doesn't necessarily take place up here in the membrane, but it can be any sorts of things like this. It can be, targeting an enzyme, it could be targeting structural protein, it could be targeting another gene. So there's lots of different ways, the signal or the cell could respond. It just depends on the message. There's thousands or hundreds of thousands of different ways that could actually take place and respond. So just a close up here, same idea. We have our hormone, which is our ligand, and it binds right here. And you can really see, kind of look like a hockey stick here for a little bit, or, person's foot. And then once it bound up here, that caused a conformational change and this thing straightened out. And now it makes it more receptible to particular reactions on the inside. So that's really how the signal gets from the outside to the inside. The ligand binds to the receptor, which is a protein, and you get some sort of conformational change or shift, which then triggers the rest of what's going to happen on the inside. So we have a few different pathways here, but hang on one second. I'll be right back. Okay, sorry, really need a drink of water. So one of the first pathways we'll talk about right here is kinase, the kinase pathway. And you'll learn that kinases do one thing. They phosphorylate things, right? And we've talked about phosphorylation a little bit, and that's when you add a phosphate onto something. And so it really happens a lot when you use an ATP, like down here in this picture. So grab my pen here. Okay, so you get, the ligand binding here, and then you get the conformational change here. And so what that does in this case is it allows ATP to interact with this protein right here. So ATP comes up here, and it does have 3 phosphates on it. But then once it interacts with it, notice you get ADP. Okay, so you only have the 2 phosphates here. The third one disappeared. And so it ends up down here. Okay, and it is on this other protein, this purple guy down here. And so because you added a phosphate to it, that protein was phosphorylated. And kinases generally do this. Kinases are enzymes, right? It ends in an ASE. So a kinase is an enzyme that phosphorylates things. And when you phosphorylate something, you generally get some sort of trigger, some sort of reaction, something that's going to go do something. Okay? Phosphorylation will keep coming up, not only this unit, but down the road a little bit. Okay, G protein coupled receptors. This one's going to take a little bit to explain. It's going to take several slides, several different types of images, so you'll have to just kind of stick with me here. And you might need to go back and replay it and relook at this, or go watch a video on it. It's not super complicated, but there's a lot of things that have to happen, a lot of steps. So here is the GPCR. Okay, G protein coupled receptor. That's this thing. It's just a particular type of receptor, and what it's going to do is it's going to deal with GDP and GTP instead of ADP and ATP. Okay, so just like ATP is adenosine triphosphate, you have GTP is guanosine triphosphate. So it's just one of the other bases, right? One of the other nucleotides. And in the ACGs and Ts, we're talking about the A for ATP and the G for GTP. So the first step here is just the actual signal, right? The binding of it. Okay, so you have the ligand that binds, and you have the conformational change. Instead of this being curved, it comes over here, and this is straight. And this allows this complex to now bind to it. It's inactive over here. Okay, but now it can bind to this, and that's going to trigger several other things to happen. Okay, so view this as like a timeline. Okay, so the timeline's kind of going across this way. So it binds, this is inactive, and then now it's going to become active because this is binding to it. And so what happens is GDP, right here, GDP gets kicked out. Okay, GDP is this blue guy right here, and he gets kicked out, and then it gets replaced right here with GTP, the pink one. So the blue one over here gets kicked out, the pink one goes in, and that's going to cause these two big complexes right here to split. Okay, so this whole thing was one thing altogether, and now we're splitting it into two separate things over here. Okay, so stay with me. That's just kind of the first part of it. If you're not sure, I'm going to keep repeating this a lot, so just stick with me, okay? There's different pictures that show you different parts of this. We will come back to that in just a second. Right now, we just did number 1, number 2, and about to do number 3. I'm going to come back to the GPCR. That was kind of just an overview of it. We're going to spend some details on it in a minute because it's kind of the longer one, the tougher one to understand. Ligand-gated ion channels, not very tough to understand. So in this case, you have a ligand-gated ion channel right here, which are these two big proteins that are sitting right here in the membrane, and they have little active sites right here. And when they're empty, then the gate stays shut, and it stays closed off like that, and so nothing's going to get through. But look at this molecule right here. So if they come and they bind to those active sites, now the channel is going to open, and now ions can get through this space, and all the little yellow ions here are able to travel from the outside of the cell to the inside of the cell. Okay, so ligand-gated ion channel. Okay, ligand-gated because you have to have the particular ligand to open the gate, and it's for ions to get through, right? Little big amounts of small molecules to get through. Okay. Second messengers we need to talk about. So there's a few different second messengers. The second messenger on this page is this right here, CAMP. Okay, so we'll talk about that as we go. So CAMP, or its real name is cyclic AMP. Okay, that C stands for cyclic. So that word right there, cyclic AMP. So AMP is a derivative of ATP. So we have adenosine triphosphate over here, 1-phosphate, 2-phosphate, 3-phosphate. We have our sugar right here, and we have our base up here, okay? And so there's an enzyme right here called adenylyl cyclase, and it is going to cut off 2 of the phosphates. So you have 3 phosphates, it's going to cleave 2 of the phosphates off, and now cyclic AMP, cyclic adenosine monophosphate, right? It only has 1 phosphate right here now. That's going to be CAMP. And then with a little water and another enzyme called phosphodiesterase, we're going to have what is just AMP, or adenosine monophosphate. Okay, the only difference there is where this phosphate group attaches. We have a little rearrangement is all that happens. And you can see, like, between this box and up here, this all looks slightly different between these two, but not huge changes. You don't even need to necessarily pay that much attention over here. Cyclic AMP is the one that we're going to spend some time on because it is going to be an important second messenger. So here's our G protein coupled receptor again. Okay, so we're coming back to this idea now. So this GPCR was the one, pathway number 2 that I started to describe earlier. So here's our first messenger, right? This is our ligand. Our ligand is our first messenger, and it's going to come in here, and it's going to bind with this G protein coupled receptor. And here is now our G protein. And remember, GDP got kicked out, and GTP got put in. And then we have the adenylyl cyclase here, which is going to take ATP, and it's going to convert it into CAMP, the second messenger. So the first message comes in and causes these first couple triggers, but then the message, it would kind of die if we didn't have the second messenger. But now ATP gets converted into cyclic AMP. Remember, by this enzyme, that's what's doing it. Okay, that's what's making this conversion. Okay, so that is causing that. And then the second messenger which is CAMP, cyclic AMP, is going to activate what we call protein kinase A, or just PKA for short. Okay, protein kinase A or PKA. So over here in these words is what is also really going on. So if you're not following too much in the pictures, then look over here. It starts with a ligand and the GPCR. So the ligand binds, right? You can find that here or here, and you can also find that right here. GDP gets kicked out and GTP in. That is going to cause cyclic AMP to be activated. Okay. Then cyclic AMP is going to activate PKA, protein kinase A. PKA can phosphorylate many other proteins to form a cellular response. So PKA, protein kinase A, what do kinases do? Kinases phosphorylate. Kinases phosphorylate so that something will then trigger and something will happen. You will get a cellular response. Okay, so that's not the last time I'm going to bring up GPCR, but we're going to keep going for now. A little bit more on second messengers. There's another important second messenger that we use, and that important second messenger is this guy right here, calcium. Okay, so signal transduction can be used accomplished using calcium ions. Okay, so in the cytosol, it's usually pretty low. So out here in the cytosol, calcium is usually pretty low. Okay, and it's high inside the ER, and it's high inside the matrix of the mitochondria. Okay, so we store them in a couple different, we store calcium in a couple different organelles. And so, as I mentioned earlier, cytosol concentration is low. If a signal binds, if you get this signaling molecule, okay, then calcium is then going to be released from the ER or from the mitomatrix. So we're going to get this calcium coming out, and it's going to flood the cytoplasm with calcium, okay? And then once the signal is gone, the calcium will then get pumped all the way out of the cell, okay? So down here, the calcium will get pumped out of the cell, and then the levels will get returned to where they are supposed to be, okay? And we'll talk more about calcium as well as we go, but just remember calcium is a good second messenger, okay? It can activate certain things to happen as well. All right, back to the GPCR. Here's our most specific example yet, okay? Now we're going to use this and we're going to talk about epinephrine, okay? AKA adrenaline, also known as adrenaline, okay? So adrenaline, you generally feel adrenaline in a couple different situations, right? Fight or flight, short-term stress. So if you get really excited, you could get some adrenaline, or if you get scared, you could get some adrenaline. And so what we're looking at, here's epinephrine, okay? This is the hormone that's coming in, and it's going to be the first messenger. It's going to be the ligand. It's going to bind with the G protein coupled receptor. GDP is going to get kicked out. GTP is going to go in, okay? Then this enzyme is going to convert ATP into CAMP, which is our second messenger. That's going to activate PKA, our protein kinase A, which then is going to phosphorylate, and it's going to, it's going to cause a couple things to happen. It's going to cause the inhibition of glycogen synthesis and the promotion of glycogen breakdown. So remember, glycogen is how we store our sugars, right? That's how we take our glucose, our excess glucose, and we store it as glycogen. And so if you're inhibiting glycogen synthesis and you're promoting glycogen breakdown, well, that's because it's fight or flight time. Okay, you don't have time to be building glycogen. You want to use all that glucose right then, and you want to break down all the glycogen that you have and flood the areas with sugars and get the mitochondria going and get that electron transport chain going and make some ATP, because it's fight or flight time, right? So you need that burst of energy right then. And so that kind of answers this question, right? You don't want to spend time building stuff when it's fight or flight time. It's time to break it down and use it. That's why you have it in reserve right there. That's why you're storing it, just in case you need it for stuff like that. Amplification. This is just kind of a quick slide. If your signal is on, right, if the signal is bound to the receptor, if the ligand is bound to the receptor and it stays there, then the signal keeps getting propagated as long as the ligand is there. So, you have the signal and receptor and all the, the GDP out, the GTP in, the adenylyl cyclase does its job, and you make a bunch of CAMP, and the CAMP is activating PKA, and the PKA is phosphorylating all these things, right? You have all these yellow phosphates all down here because everything got phosphorylated, and that's triggering all these these purple guys are the target proteins. They're triggering these proteins to go do their job. So the longer this stays, look at the amplification of that signal. All of this work, okay, all of this work down here, all of this cellular response is coming from one little signal. So you can amplify stuff very quickly this way. Cascades are another way this can happen. So I do not want you to memorize all of these steps. Do not want you to do that. But the idea is you have a signal molecule, and if that signal molecule stays there and this stays activated, okay, then here's your first protein right here, and you have the PKA, and it's going to go activate here, and then this one's going to go activate, and you have this cascade of reactions that are going to go on until you get the cellular response down here at the bottom. Okay, so again, this would keep happening as long as that signal stayed there. So don't worry about the actual names or anything like that of this. Just know that this is how a phosphorylation cascade happens, where you keep breaking down ATP and you keep phosphorylating the next protein, and it's just really a chain reaction that's going on. So what kind of feedback is this? This is the same idea, right? We have our three phases. We have reception, we have our transduction, the signal being sent into the cell, and then we get our response. Well, if everything keeps going this way, right? If everything's going this way, is this positive or negative feedback? Is this more get you less, or is this more get you more? Well, the longer it stays on, the longer you bind to this, the more of this you get, the more cellular response you're going to get. And so this would be positive feedback, right? More gets you more. Simplicity leads to complexity. There are also sometimes our one signal here can cause different things to happen. So you get your conformational change in your receptor down here, and our scaffolding protein now, scaffolding because we have three different protein kinases on here. So we have three things attached to this pink one, which is called the scaffolding protein. And you can tell this little notch right here and this little notch will actually, shape-wise, they will be able to bond. So when those two bond, that's going to trigger three different protein kinases. They're all going to go off and do their own jobs, and you can get three different responses from the one signal right here. Another example of this, of simplicity leading to complexity. So a few things happening here. So signal molecule binds right here, receptor, transduction, relay, and you get one response. Okay, pathway leads to a single response. That's your typical one. Over here, you have a ligand that binds, and now this guy activates two separate proteins or molecules in there, and so we're getting two responses. So in this second pathway, cell B, we're getting two responses from one signal. Okay, so that's a different way it can happen. It can also happen like this, where it takes two signals to generate one response. Well, why would that be? And we call this crosstalk. Crosstalk occurs between two pathways. So it takes both of these pathways to make this one cellular response happen. And the way I kind of relate this is like submarine, like a nuclear sub. If you watched movies where they do that and they want to launch a nuclear sub, is there just one key for that? Generally, it's a two-key system, right? And the keys are put so far apart that one person could not physically stretch their arms and do both keys. It's not physically possible. They're like 10 feet apart. So you can't do that. It takes two people to agree that yes, we should launch this missile. So in this case, it takes two ligands to say yes, we need this pathway right now, because it might be a pathway that can also cause some damage if it's not being run properly or if it's left on too long, something like that. And then the last one here, up here, notice it's the same ligand. It's the same signaling molecule as the first one, but it is not the same receptor. Okay, I'll wait for this is a different shape than this over here. So same ligand, different receptor, and look, this is different from this, and this is different from this. So just by having a different receptor here, it sends it down a different pathway, and you get a different response as opposed to this one. Okay, so that can definitely happen as well. A different ligand with the same receptor, a different receptor with the same ligand. All these combinations can lead to different things, which is why it's so complex. Cell signaling is a very complex process. And then a more specific example of this is apoptosis. So we've talked about this before, programmed cell death. So if we look in here, a couple of the important things. So here's your receptor right here, receptor for death signaling molecule. But in this one, in diagram A here, no death signal. So we don't have a ligand, and this is how things are organized. You have this protein SED3 and this other one called SED4, and they are being inactive right now. Okay, and then SED9 is active. SED9 is active, and it's sitting right here on the mitochondria, and it's basically blocking SED4 from binding to the mitochondria. So when we go over here and we look at the next diagram, which has the death signal, here's your death signaling molecule, and it comes in and it binds to the receptor, and that causes this pathway right here to then inactivate SED9, and SED9 is going to leave and get out of the way, and that's going to allow these processes to happen. So now SED4 is going to be active, and SED3 is going to be active, and they have this cascade, and they are going to activate two things, proteases and nucleases. So what do you think proteases will act on, and what do you think nucleases will act on? These are proteins that are aces that are named for what they work on. So proteases break down proteins and nucleases break down nucleic acids. So if you're going to start breaking down nucleic acids and proteins, is your cell going to survive? Probably not. That's your apoptosis. That's programmed cell death, right? And then this question up here, why are death proteins in a non-apoptotic cell? This cell is not going through apoptosis, but it still has the death proteins in there. Well, why would that be? Well, it's because if they weren't there and you needed a cell to go through this, what would have to happen? Well, you'd have to have DNA to RNA, sorry for the writing, all the way to a protein, right, in order to make a protein. So it takes time to run the central dogma, right? This would be the central dogma, and we'd have to find the instructions on the gene and copy that, and the mRNA would have to leave and go find a ribosome and start gathering amino acids and building the protein, and all that takes time, where most of the time, if you want a cell to die by apoptosis, you want it to happen quickly. So these proteins are in here dormant. It's just, this is kind of the brake pedal. The SED9 is kind of the brake pedal, and if the brake pedal gets removed, then those things are off and running, they're doing their job, and they're going to start breaking down the cell. Okay, so that is cell communication. That was our last set of notes for the cell unit, and hopefully that made some sense to you guys. If not, you're always free to ask me some questions. Send me a note to the Canvas inbox. We can jump on a WebEx, whatever you need, and you have some practice questions right here to deal with if you so choose. Take care, guys. Talk to you soon.